so he has an interesting history. He's right now a professor in the University of Grenoble, but he's also spent a lot of time in Montpellier, also working with the PlantNet group there and the French Institute of Moderator in India, where he was a research head of some sort for 3 years. And what I think is really nice about his work is that he uses a bunch of different tools and approaches to link between community ecology and broader scale macroecology and even macroevolution. And this, I just think, is always a challenge that we all face. And he has some really elegant ways of trying to connect these scales. And I look forward to hearing more about this. So thank you.
Thank you to you. And I'm very happy to be here. So I have a wonderful discussion with some of you yesterday and today, and I will hope to continue after this presentation. You have a very broad title, because in fact, what I will expose is a framework we are developing currently, and with different questions we can address with this framework. But the important point is that we are working on eco-evolutionary dynamics. That means that we want to meet different spatial and temporal scales to answer a question in macroecology and historical biogeography. Just to show and complete my profile, I was first an engineer in water and forest management. After that, I did a PhD in ecology. And after, I went to University of Montpellier first, and University of Grenoble more recently. And that is interesting also to mention that we have this institute, French institute in India. It is one of the rare foreign institutes in India because it's a very close country. And we have interesting resources. We have a botany lab, we have a palynology lab with a lot of data. And we are managing a permanent plot, which for more than 40 years now. And we see many interesting things with this plot. So about the question and research topic, I work on different aspects, different kinds of models at different scales, on community dynamics, local community dynamics, depending on the coexistence and the interaction between organisms and their environment, but also in the fact that you have a network of suitable habitats that make meta-population or meta-community. So I work on this kind of approach. And then the new question that emerges and we'll explain today is to connect the dynamics over ecological scale with the dynamics over evolutionary and biogeographical scale. So I will present both theoretical and empirical approach to try to infer, to analyze patterns, and to see which kind of mechanistic model can be relevant. And also I work with the national park and reserve manager to try to have some tools to help them to, with PlantNet especially, tool for using for management and for information. Okay, so the big picture is there is a hierarchy of processes. And I think all you know this kind of picture. You have many, many ones. When you have a kind of big pool, which is a regional species pool, and you have a set of filters. So you have first the dispersal and chance filter, so meaning that you need to disperse and establish at a given place. If you can establish at a given place, it will depend on your adaptation to the abiotic environment, so it's environmental filtering aspect. And after, in the community, you have the internal dynamics of community with interaction and connection between communities. So that is the pattern of diversity within and between communities that we try to decipher at the end. So I introduce also the idea that we have multiple facets of diversity, and I will show after that different facets are useful to disentangle the ecological and evolutionary dynamics. You have taxonomic, functional, and theoretic diversity. Functional diversity is mostly used to see the influence of functional traits and age differences on the composition of community. Taxonomic diversity is interesting to assess the demographic and dispersal-based limitation, especially with the neutral theory. We derive some expectation in neutral models on alpha diversity, beta diversity, and with different phylogenetic resolution. And phylogenetic diversity, also we use it to see how we have a legacy of some niche conservatism, or on the contrary of diversification of species in different kinds of ecosystems. So the topic today is very broad topic, is to connect what happened over a long time on the biota and how the spatial and temporal dynamics are driven by the environmental change. There was huge environmental change over the long term, and we may try to reconstruct with data available what could be the journey and the diversification patterns for these organisms. And I will start with an example because I work in India, which is a journey of Madagascar and India, which are both part of Gondwana. And at some point, it has separated from Gondwana, but staying together for some time. And at the end, Madagascar and India separated, and India continued until Eurasia, with the Himalaya, rising Himalaya. So you can have some time period which represent different states of configuration in terms of dispersal limitation, in terms of habitat suitability over time. So we can try to link this information with actual biodiversity. So just a simple example, we have done this kind of analysis in phylogenetic diversity. So you have two colors. One is for India, one is for Madagascar. And what we do is that we take some specific time steps which correspond to different periods of the geological and climatic change. And we just are counting the length of branches which are related to tips of one or the other of the two regions. For instance, you see that you have one branches. So do you see this one? No. Here, or this one here, which is shared between ecology and India, and Madagascar. So you can sum the lengths of the branches, which are unique to one of the two countries, or shared between the two countries. So you can do that for each slice of phylogenetic trees. And we did that for different periods, which are very important in the history of India. And you can see that the relative proportion of only India, only Madagascar, and shared diversity is changing over the time. And you can investigate why and how this contribution of phylogenetic diversity is different between India and Madagascar to interpret their diversification pattern over the long run. So for instance, we're using a huge null model where we sampling the location of individuals in India or Madagascar. We can see the deviation from a null model for each slice of the phylogenetic tree. And we see, for instance, the first one, the effect size is high on a recent period, and it is low in older period. And if you look at Madagascar, it's the reverse. It is very low at recent period and very high at older period. So it's not neutral. It's not null model rather. And we can assume that we have a museum effect working more in Madagascar, while India has undergone big diversification after the collision with Southeast Asia. So that's an example where we can find different patterns, this pattern-oriented analysis, to discuss and decipher what can be the different driver of diversification. I talk also about historical biogeography, and we are especially interested in historical biogeography by the role of dispersal and vicariance. And this is discussed a lot because some are promoting more vicariance, some promoting more dispersal. So it has discussed a lot. And connected to this historical biogeography thing, you have people working on phylogeography, which is in fact a way to analyze the events of fragmentation, the migration pathways, the population dynamics, based on the current genetic structure of populations. So they do some inference of the most plausible scenarios relating the population between them and their migration in new area, for instance. So just a very simple example, we can imagine that in a set of islands of different age, you can have different scenarios of local endemism, which is related to the age of the the island. So you can expect that it is reflected in the size of the branch in the phylogeny. And you can have sometimes backward migration, for instance, in the bottom image, which are related to some immigration events at some point. So the idea is that if we consider the spatial structure of the phylogenetic diversity, we can get insights into the evolutionary aspects. So we work on this topic, so it is part in French, and I could not edit it. But in fact, it's very simple. We consider all fluctuation of environment, which are represented by the spiral, and the tectonic dynamics, especially for tropical forests, the separation of the Mont Gondwana, can be linked to ecological processes on population dynamics, and over the long run on apparition of new species, speciation and extinction in evolutionary time. And the question is how the different environmental change in the past at different scales have shaped the diversity of species in the actual current habitat. And for that, we use something which is common in phylogeography, which is a coalescence-based approach. Coalescence-based approach is a way to reconstruct the shared ancestry of individuals in an actual present-day community. So here you imagine that you have some trees, sample in the community, and you know, for instance, in this case, that they are sharing some ancestor at different times, and different branches are merging together. And we have a lot of theoretical analysis. We connect the way of branching in this tree, a genealogical tree, to the parameter of drift, genetic drift, and migration pathways. It is commonly used in phylogeography, and for instance, you can imagine that you have a first phase between different groups of population, and then you can have a diversification which is related to the isolation of different regions. And people use that with population genetics tools for investigating many species. So when we talk about comparative phylogeography, some people try to compare now the phylogeographic pattern of the different species, which are here some species in Asia and Oceania. And you can see that we have both interspecific range, which are different, and intraspecific diversity, which is also expressing the barrier. In fact, it is close to an effect of the Wallace line, which is related to the same driver, long-term driver of separation and limitation of migration between the species and between the population within species. So that's not new, but that's done for each species separately. And what I propose is to do a multi-species coalescence, so that, in fact, we have a single genealogy in the whole actual community sample. And we try to incorporate their shared ancestry, but also their shared other species, related species. For that, you consider that in the genealogical tree, you can have some speciation events, and then all the descending individual of these speciation events represent a current day independent species. And so we can use an approach to model the dynamics of population within communities, with dispersal and with demography, and to connect it in the past with the events of speciation, which were linked to previous habitats. So the big point for doing that is that we need to transform the genealogy of individuals to a phylogeny of species. So I can go back. So here, for instance, we use a model developed by the Hélène Morlon team in France, Manso et al. In Ecology Letters. They consider that you have some rules, you have mutation and genotypes which are appearing after mutation. And if you have some monophyletic group descending from the same ancestor and over a long enough time, they consider that it is a new species. You have different speciation approach, but this speciation is very simple. It is a protracted speciation. You have a set of different genotypes, and at some point, it establishes a new population without breeding with others. So we can assess the diversity of actual community, current community, with the diversity of their phylogenetic structure, and also the other facets of diversity. So here. So what we want to do, it is to put together the picture of the changing environment over the long term, in terms of climate, in terms of tectonic events, and the events of coalescence and speciation in the big genealogy of individuals. So PhD students that they funded last year work on software to compute this big genealogy over reasonable time using the coalescence-based approach. And when we can simulate different scenarios with different genealogy, you can have a broad spectrum of patterns with different values of parameters of speciation, mutation, and drift. And you can test hypotheses about which kind of events, for instance, the separation between the two continents, at which time the migration has been limited. If you have change of population size, you can see the effect of refugia and small population bottleneck at some time. So what we do is using the coalescence-based approach to do an inference of the parameter values in the eco-evolutionary model. So you start from observed community. So I talked about that in ecology yesterday, but we use that in an eco-evolutionary way also. So you have a number of observed summary statistics, which are selected to reflect different aspects of actual recent dynamics and long-term phylogenetic structure. And we compute functional diversity also if you want to take account the effect of niche-based processes. For each parameter of the assembly model, eco-evolutionary model, we have a prior distribution of parameter values. So we can, for instance, have a uniform prior distribution for parameter value of dispersal and for parameter value of habitat selection. And then you simulate the genealogy and the composition of the community that can happen with different values of this parameter. We select the most similar patterns. So for instance, the second pattern is closer to the observed communities than the two other ones. And based on this distance between the simulated communities and the observed community, we can derive a posterior distribution of the different parameters of the evolutionary dynamics. This kind of things is done a lot in phylogeography, but we want to do that in a multi-species setting for multi-species coalescence. So there is a lot of things to check because we have a lot of hypotheses underlying this kind of approach. And there is a lot of debate, for instance, about which role vicariance and which role migration can play to structure the biota in different continents. What we can do is to use different models and to make competing them, observing simulation of the two models and see if we can allocate the simulation rather to one scenario or rather to another scenario. That is a misclassification analysis. And because you have a lot of debate about what is the signature of processes over the long term, it's useful to know if the model is able to differentiate contrasted scenarios with very different history of migration and vicariance. So that's the first point, is to ensure that the conditionality bias, the fact that we have selected a model with all these parameters, don't prevent us to test the model against another one. Another aspect that we use before trying to apply to data, it is a cross-validation analysis. So in this analysis, you take one simulation as it was observed community, and you compare this one to all the other communities, and you do what you want to do, inference of parameter value. And the line represents the perfect fit between the true value and the observed value. And if it is very noisy, that means that you have some uncertainty in the estimation of this parameter. So we did that for a different eco-evolutionary model, and we wanted to get enough information in the summary statistic to be able to separate and identify the parameter value, each parameter value. Something what is useful also, it is a posterior predictive check. So you have selected a set of parameter values as the most plausible one to explain your pattern. And you can simulate other aspects of diversity, like beta diversity, for instance, and see if it fits with the data. So it's kind of posterior predictive check. Is the selected model able to reproduce observed biodiversity pattern? So you see the different stages are very important in order to decide if we test, validate some hypothesis, and we can be confident in the parameter value we get. So in fact, these students was very interested to summarize by a big inference framework. You start with all the aspects of model design. You simulate the coalescence with some constraints over time, like the changing population size, migration between regions. And if you have two models, you try to compare them and select them. If you have one model, you do the cross-validation and the parameter check, posterior check. So that is what we have done in the EcoPhilo project. So we have a package in Python language, which is used to simulate a number of samples in ecological meaning. And you have a function to do many simulations. So it is to batch a simulation with parallelization and things like that, in order to simulate many different parameter values. So the idea is to use ABC in eco-evolutionary model analysis. So the next point, if it's okay for now, next point is to see where is the interesting information and is it relevant to use that for inference. So we tested, in fact, for instance, the effect of habitat reduction, which is a common constraint in the past dynamics of communities. So bottleneck, which is more or less narrow or more or less old. And we just see the effect on the coalescence and on the observed abundance and observed threat diversity. So we use different values of bottleneck and different values of mutation and threat evolution to test what is the signature in present day of the past bottleneck. So you have different scenarios and you have different way of reducing or expanding population over time. So for instance, if we look at pattern of taxonomic diversity, like number of hills, Simpson index, etc., we can see that if we consider the recent expansion dynamics, like 100 generation time, you can find that there is a connection between the reduction of habitat with more extinction and the species richness. So if it has been very restricted, you have less species. Okay. You have the same if you look at bottleneck, which are older, but it is vanishing at some point because you cannot relate taxonomic diversity to very old events because it has been in equilibrium with new conditions and you don't have any signature anymore of the past habitat reduction. So it's a way to see, do we have a signature on taxonomic diversity for different age of the phenomenon? So we did the same for phylogenetic diversity. And here it's interesting because we can keep track of older events on different aspects of phylogenetic diversity, mean phylogenetic distance, most recent common ancestor, and nearest taxon distance. And then you can see that with some confidence, you can connect the different variation of phylogenetic diversity to the expansion and the reduction and expansion time and amplitude during the expansion. Is it clear? This okay. We worked also on functional diversity with threat evolutionary aspects, so to see if we can have the testimony of diversification which happened before or between before within or after the reduction of habitats. So it's especially useful to set hypotheses about apparition of original strategies, which can happen in a remote line, which has only survive a few individuals. Here, so what we see is a parameter of variance of strat evolution, sigma, and the speed of decreasing evolution, meaning that if it is minus negative, beta is negative, it was diversified long time ago, and after it's slowing down. If it is positive, it diversifies very late in the process. So we can see that according to the scenario, we don't have the same signal on phylogenetic variance, on functional variance, sorry. And especially in this case, what we can see is the effect of recent trait evolution. So that's first work is a way to simulate like a theoretical experiment, the patterns, and to see what is the linkage. Can we link the patterns with different parameters? And now the project, the ongoing project, is to do that for tropical rainforests, which are fascinating for their high diversity and their low abundance of population locally. And especially with testing this Madagascar and India scenario. And we have a lot of sampling plots in Madagascar. We have a lot of sampling plots in India. And we want to establish scenarios like that. Scenarios are kind of the simple representation of the changing of habitat size and the separation of different habitats across islands. And we can define the timing of these events. So what we do is coalescent, multi-species coalescent for this kind of scenario. And we want to investigate if the separation of Madagascar and India, at which time it can have an influence in the model, and at which rate there was some migration events after the separation between the two. So we will use, in fact, a set of forest plots that has been followed and sampled in different continents. So this is all map of rainforest in different continents. And there are a number of available data sets. One also in French Guiana, which is managed by colleagues of Montpellier, with different information about abundance pattern. This is abundance pattern. Il veut pas. Non, c'est pas ça. Okay, this is good. You have abundance pattern, you have genetic diversity information, and you have trait information. And we want to investigate summary statistics of their diversity in different forests and to analyze our ability to infer the effect of past environmental change and migration pathways. We will do that also at the scale of Amazonia. Amazonia is covered by a rainforest, but it has a historical change of savanna and forest elements during the drier period of the Quaternary. And we want to see if the classical hypothesis about the refugia in Amazonia can meet our expectation in an eco-evolutionary model. If we see the different fragmentation events and the migration limitation. So we work with a ATDN network, which is a huge data set in Amazonia. Plenty of papers have already been published about pattern of diversity, but no paper has tried to use a mechanistic eco-evolutionary model on that. What I can show is first results. So first results, we have taken only South America and Africa, and we have set scenarios of changing size in the past, according to a time scale here. And what we can see is that in America and Africa, the best posterior distribution of the effective size is the same pattern. There is lower effective size during the last glacial maximum, for instance. Meaning that if we take together the information on taxonomic and phylogenetic diversity, we can see that there is a signature in the response of this species to the previous reduction of habitat during the AGM, which was a far smaller cover of rainforest in this time. So it's the first result, which is quite encouraging for us. We will go ahead to analyze more precisely which metric contributes more to explain that. And we want also to increase the data set to see if it is robust to our sampling design. And that's what is actually the EcoPhilo project. So the EcoPhilo project, you have an available tool in Python language. So it's available on GitHub. So if you want to use it, you can simulate using the multi-species coalescence. And now we also want to tackle different challenges. We want to encode the spatially explicit coalescence. We have some tools, but they are not very quick and easy to use. We want to incorporate selection in coalescence. You have different kind of coalescence according to your selection and interaction between species. And after, we will also evaluate our capacity with the misclassification and the cross-validation things, the predictive check, to see if with a model and some information on the data set, it's enough to infer what is happening in the eco-evolutionary time scale. So it leads also to select different facets of biological diversity using both phylogenetic and intraspecific genetic diversity to see the connection between the two, and to integrate also fossil data to define the fact that at some point of the time there was such diversity of families, for instance, in India. We know that, and we can constrain only the scenarios that predict these kind of things. So the more interesting data we have in the history, the more accurate should be the estimation with the distribution of ABC. And I have a parallel of this approach in eco-evolutionary dynamics with another tool which we developed, which is Ecology, which is more for community ecology, community assembly and environmental filtering. In this way, you also have the pool of species, you have the genealogy of individuals in the community, which are descendants of migrants. The migrants are established according to their the fit of their functional traits to the local environment, like is shown by this probability curve. So niche-based filtering has happened. And you can adjust parameter of migration from this pool, parameter of environmental filtering, and to have different patterns of genealogies in the community. And so we use it with a good result on cross-validation in the context of ecological systems. That's something I explained yesterday. But that's not the point today. So if you want to test ecological hypothesis, you can do Ecology with a lot of tools to do that. And EcoPhilo is more for eco-evolutionary long-term dynamics. So I'm not very long. That's all I wanted to tell you. Thank you.
Thank you very much. That was a lot to think about. I'm sure there's some questions and thoughts, and maybe also if somebody online has a question that you can let me know. So the floor is yours.
Yeah, thanks. That was really fascinating. I was wondering, and I have to admit that as you know, I don't know coalescence modeling, but do the parameters that you're estimating, are they allowed to vary in time themselves? Because the patterns that you found in the Americas versus Africa were really fascinating, and it made me wonder how, whether or not the parameters are static and how that affects the inferences, especially further back in time. Yes, and you mean also what kind of constraints we have in the prior? For instance, the MRCA has been constrained to be not older than angiosperm age. And we can check which phylogenetic diversity pattern is contributing to estimate the changing size at different period. The definition of the period is quite arbitrary. We have taken that from geological and climatic data, the relevant period with some regime, environmental regime, which can be considered consistent during this period. That's the scheme with this kind of things. We do some time slice and with different parameters of effective size.
And after, in the phylogeny, we decompose also over the phylogeny at the different time scale, and we compute the distribution of the lengths of branches at different time in the phylogeny, and we use that as summary statistic.
For each of those squares, sorry, for each of those chunks of time that you're showing there in blue, you can sort of re-estimate all the parameters in the model.
You mean for, yes, we can use only the present day data and the phylogenetic diversity, and also fossil data to set that some families were present. Even families which were very recent are present in both Madagascar and India, very common. So we need to see what scenario can explain the fact that very recent families are present in both islands while they are separated for a long time.
Thank you.
Thank you. If you go a few slides further to the back of the presentation, we had this curve about habitat availability, I think it was.
This one?
No, the curve of was it America and
Africa?
Africa, huh?
Yes.
Here we go, yes. And we see this depression during the LGM.
Yeah.
Here, and I mean, this is clear related to habitat availability, but what we see in the simulation of 7 important tree species across Europe is that effective population size remained surprisingly constant for way beyond the last glacial maximum.
Yes.
Even though habitat was also reduced, but diversity, genetic diversity, and derived effective population size was rather constant. So how does that match according to your framework?
Yes. What we get is the ancestor distribution in the different times, time of the coalescence. And we can so assess, for instance, the diversity of large taxonomic groups like families, orders of different groups, and see the pattern of diversity at this level, but it is coarser and coarser when we go in past. So we cannot investigate the number of extinct species at this time in the region, or the dynamic of colonization, like for temperate trees, which is ongoing currently. For that, we need to have some more explicit forward in time simulation. I don't know if it's a good answer.
So this actually is not this the species number curve or a taxon number curve here that you present.
Yes, what we have is effective size. In fact, you consider that the global number of individuals is constrained by the availability of habitat. So for a rainforest, it is a global number of trees. It has been estimated, number of trees in Amazonia. And in paleoecological data also, there is some estimation of the number of individuals of trees in rainforest at different times of the past. So we can correlate the estimation of the size in the model with the actual reconstruction of vegetation. That's what we want to do.
So it's a level of integration higher than at the individuals.
Yes.
That's what I didn't capture here. Okay, thank you.
But for the short, because it is short for compared to evolutionary events, it's a fluctuation of abundance also, which play a big role, because there are not so many generations between the last glacial maximum and now for this species of trees. And so you can still have transient variation in abundance related to the demographic drift. So when combining the long-term information of phylogeny and the shorter-term information of relative abundance, you have complementary picture to see what is the story.
Thank you.
I just wanted to follow up on that. So is your hypothesis then for Europe, or his hypothesis for Europe, that you would see a straight line if you were to do this in Europe?
At least within species, yes.
Yeah.
That's quite fascinating. No, sorry. So I have a question. I don't know if somebody else does. So just on that same line, if you had a fossil series, a really good fossil series, I'm thinking Chile, could you actually reconstruct this like do the coalescent and pretend you don't have a fossil series, but if you had a really good fossil series, you could have another way of testing it or looking at it. Have you tried anything like that?
We wonder how to include fossils because we don't have complete assemblage in past. We have some occurrence of species. What we can do is to select in the simulations that said that at this time there was enough lineage, this group of a group which is more common now, for instance.
So you can set it to sort of check, or you would do that as a prior?
Yes.
Okay.
And we use beta diversity information, both in taxonomic diversity and phylogenetic diversity. And so if we have some insight from fossils that some old families were present in India, you have also some fossils in Madagascar that there was some parents of Indian plant. We can incorporate them to discuss the scenarios and to constrain the simulation.
But I guess the tropics might be the most challenging place to try to do that.
Yes, but it's well adapted to do that with a neutral theory, because it starts from neutral theory, but with constraint on the habitat size. But anything else is neutral. So the coalescent is a Kinman coalescent, which is usually used for neutral dynamics of migration drift in population genetics.
I guess following up on that, in the fossil record, we tend to see just the most dominant taxa.
Yeah.
And so how does the patterns and the most abundant things in the community match the total community?
Yeah, that's something we can infer. Yeah, because at a different time of the past, you know that different set of lineage will be common, different other set of lineage will be uncommon. So you can also have a relative abundance of the different clades at older time.
So I have a question that kind of combines your approach to say the approach of Loic and you guys. So if you're doing the proper simulated model, right, you could actually do a proper mechanistic model where you put all the mechanistic stuff in there and you sort of get a hypothetical answer. Would that make any sense? And then do the coalescent back as really a way to test it?
So as far as I understood, the idea would be to have, for example, a spatial explicit model that simulates the evolution of a clade, for example, in the tropics. I don't know. And then at the end of simulation, as an outcome, you would have a phylogeny. And then could you use your method to try to reproduce this reality that you know because you simulated it in a simulation model?
You mean the past events?
Yeah.
Yeah, we think it will be reflected if we look at the distribution of branches lengths by period, like I exposed in Madagascar in India. These patterns can tell a lot about the events at different periods. So it's decomposition of the phylogenetic diversity by periods in the past. You have different ways of doing that. It is included as summary statistic. And it is a way to have the testimony of diversification events with heterogeneous rate in the past. But we need to discuss a lot after because of course, a lot of limitation. But if you find some clear pattern, at least you can say that we can have some signal of specific reduction of habitat, for instance. We sometimes with Sébastien Laverne, we think about using that also to see the dynamics of sky islands in the Alps and the refugia, all the refugia around the Alps. Maybe once I will try also to investigate that on alpine plant communities with my colleagues. So it's very different. But I was in India for several years, so I like tropical rainforest.
Are there additional questions?
I have one dumb question about the parameterization of the model. I am not sure I got it. Like, how many parameters do you have? A lot? Like, does, like, for example, the mutation rate, is it species specific? Or is it like one value for all species?
Mutation rate is you have a prior distribution for mutation rate. And in fact, here we have seven parameters of change of size for the different periods. Like I exposed before, where is it? It was a long time ago. Here, no, it's the beginning. Yes, we take the slice here, the period with very important events, and we use this as a parameter of effective size in the coalescent. So meaning that during some period, you have a broad extent of tropical forest or not, and it's influencing the effect of extinction and ecological drift.
After the parameters are not species specific within each period?
No.
No. Okay, they stay the same.
No.
And just following up question. The traits, when you study functional diversity, so the traits are the ones that are measured now.
We won't try to do that. But we have a lot of discussion about the way you simulate functional traits and phylogeny. I shown an example where we have different model of trait evolution, but it does not fit well the phenotypic space of many organisms. So we need to think about how traits are involved in the diversification, and it changes a lot compared to this coalescent if you incorporate selective pressure and specific traits, it changes the structure of a coalescent a lot. And these points at this time it's not included in the tool.
Okay, so you let the trait a part of the, okay.
In fact, we can model the trait evolution independently of the process which structures the genealogy.
On your coalescent, you mean first estimating the coalescent tree and then estimating the, okay.
Yeah.
So at some point you were mentioning that as you go deeper in time, you're losing signal.
Yeah.
Is it also the case with this method? So there you see a clear signal of the glaciation. Do you expect to still see something further back in time? Or
you mean very old time?
I don't know. I mean, 30, 40, 50 million years ago. Or the phylogenies are basically too sparse to say anything.
That's an important point. In fact, here, the period, we have heterogeneity in the limits, in fact. The period are shorter over the recent period, and they're getting larger in the older period. So that we can have enough information if we have a long segment of time on the phylogeny for the most oldest lineage. And for the most recent lineage, you don't need to have such a broad time. So we use smaller time events. So we use increasing period in past time for doing some balance of information and inferenceability.
Any more questions?
And thank you all for being here. And
Everybody's hungry. So we want to thank you again.